Good morning to all. In today's class, we are going to see about direct and indirect gene transfer methods in plants. All stable transformation methods consist of three important steps, namely delivery of DNA into single plant cell, integration of DNA into plant cell genome, conversion of transformed cell into a whole plant. So for this, we need to have a regeneration protocol for that particular plant species, or in otherwise, we need to have a plant tissue culture protocol for that particular plant species. Coming to the tDNA transfer mechanism into the plant cells. So in this, the genes present in the Vir A region are most important. They are Vir A, B, G, C, D and E. And also this tDNA that is flanked by left border and right border. So in this tDNA region, we can insert our gene of interest so that while transferring tDNA from the agrobacterium into the plant cells, our gene of interest will also get transferred into the plant cells and integrated into the plant nuclear genome. So we are going to see in detail about how this transfer takes place from the agrobacterium into the plant cells. So each gene is having a separate role in the agrobacterium as well as in the plant cells. Coming to the Vir A, it is a phenolic sensor kinase. It is a part of two component system with Vir-G, and Vir-G phosphorylates and activates the Vir-G. And whereas Vir-G, that is a transcription factor responsible for activation or induction of all other virulent genes. Whereas Vir B1 to B11, that is a component of membrane structure, that is for transfer of the tDNA process. And Vir C, that is a overdrive binding protein, that is enhances the efficiency of tDNA transfer process. And Vir D1, that is required for tDNA processing and modulates Vir D2 activity. And Vir D2 that mix the tDNA in the right border region, and that will direct the tDNA through the Vir B and Vir D4 transfer apparatus or the pipeline. And Vir D4 that is a component of transfer apparatus, and whereas Vir E, it is specifically Vir E1, that is a Vir E2 chaperone, that is required for Vir E2 export from the agrobacterium species. And Vir D2 that is also having a function in the plants, that is this Vir D2, that is that's role is nuclear targeting of transfer DNA, and as well as protects the 5' end of the tDNA from the nucleases, that is also involved in the integration process. And coming to the Vir E2, that is having a role only in the plant cell. So this Vir E2 is known as a single stranded DNA binding protein, and that will prevent the tDNA from the nucleus, and as well as involved in the nuclear targeting and passage through the nuclear pore complex. And also it directs the protein coating T complex, and also protects the tDNA from the proteasome complex. And whereas Vir F doesn't have any role, whereas Vir J, that is also important in the tDNA transport or export. That's what here this table depicts the role of this Vir region, that is the protein encoded by the Vir genes, namely Vir A, G, B1, B11, C1, D1, D2, D4, E1, etc. That is having a function in agrobacterium as well as in the function in the plant system, that is given in this table. So here how this agrobacterium could be used as a tool for plant genetic engineer. So here, that is the problem here is agrobacterium as such, it causes a crown gall diseases or the tumors. So how we can re-engineer the TI plasmid to make it useful for our gene transfer methods. So for that we have to delete auxin and cytokinin gene by retaining the vir genes as well as left border and the right border as well as the origin of replication. And the TI plasmid, that is huge, that is 120 kb. So we have to make into a small plasmid. So here we can, so by for making the TI plasmid into a small plasmid, so we can have a binary vector system, that is vir gene and tDNA, that can be on a separate plasmids. So in the case of TI plasmid, it will have only the vir genes in a separate plasmid with the origin of replication. Whereas in another thing, that is a binary vector, so this binary vector, that means binary vector means it will have a origin of replication in E. coli as well as for the origin of replication for the agrobacterium. So that means this vector could be multiplied in both the bacterial system, namely in E. coli as well as in the agrobacterium. So apart from this, in this binary vector, it will have a tDNA region flanked by the right border and left border, and this in this tDNA region, it will have a cloning site for the plant genes as well as selectable markers. So that could be used as a marker system to select the transformed cells in the plants. As well as in the outside the right border and the left border, we are having a selectable marker. So that selectable marker will be used to select the transformed bacteria. So here, so vir genes and the tDNA will be on the separate plasmids. Only the left and right borders are required for the tDNA to be transferred. So that is flanking the tDNA region. So coming to the steps in the plant transformation, namely with the agrobacterium mediated plant transformation. First, we have to propagate the binary vector in E. coli. So that's why already we have seen in the earlier slide, it is having the origin of replication for both the systems, namely for the E. coli and the agrobacterium tumefaciens. So that this, using this origin of replication, this tDNA vector or the binary vector will multiply in the E. coli system, then isolate the binary vector from the E. coli and then engineer with the foreign gene, namely the our gene of interest. So this our gene of interest may code for anything, that may be code for insect resistance, disease resistance, or for quality improvement. For example, in India, we are having a commercialized ABT cotton, so we can clone this, that is cloned with the Cry1AC gene in case of Bolgard 1, whereas it is in case of Bolgard 2, this cloned with Cry1AC and 2AB. So then reintroduce the engineered binary vector into E. coli for amplification. Then isolate the engineered binary vector and introduce into the agrobacterium containing the modified small TI plasmid. So this agrobacterium will contain only the small TI plasmid. So we can engineer or introduce this engineered binary vector containing our gene of interest into the agrobacterium. So in order to introduce our engineered binary vector into the agrobacterium, we are having the methods called electroporation or triparental mating. And then after the triparental mating, we can engineered this agrobacterium could be used to infect the plant tissue, so that the engineered agrobacterium will be used to transfer the plant cells. So after plant transformation, the tDNA gets integrated into the different sites of the nuclear genome. So in that case, each cell is hemizygous for the insertion of a foreign gene. That is, only one of the homologous chromosomes gets the insertion of foreign gene. So the consequences of insertion are, that is a foreign DNA, that is our gene of interest, gets inserted, and thereby it leads to insertional mutagenesis. Thereby, due to insertional mutagenesis, it does not kill the cells, but at the same, because the organism is diploid. So coming to the components of transformation vector plasmids, it must have a border regions, that is the tDNA flanked by the left border and the right border, and among that the right border region is the most important one. And we need to have a selectable marker gene. So we are having a kanamycin resistant gene, that is NPT2, that is neomycin, codes for neomycin phosphotransferase enzyme, and the NOS promoter, polyadenylation sequence, hygromycin resistant gene or a HPT gene, BAR gene, that is resistance to herbicide phosphothioxin, and DHFR gene, that is resistance to methotrexate, EPSPS gene for resistance to Roundup herbicide. These are all the some of the selectable marker genes used for plant transformation. And also we need to have a scoreable marker gene in our construct, binary vector construct, but it is an optional thing, but it will be very useful. So like neomycin phosphotransferase activity, that is by as well as by use of opine production, or the most commonly used scoreable marker is a Gus gene, that is a beta glucanase activity, or green fluorescent protein, or luciferase activity. So among all these things, this Gus activity is the most important one, or the widely used scoreable marker gene. So by which you can easily screen the transformed tissues versus non-transformed tissues. So here in case of by assaying the Gus activity, the transformed tissues will turn into blue color, whereas non-transformed tissues will be white in color. So by the way, we can easily score for the presence or absence of the transgene. So here, and apart from that, we need to have a cloning site for the gene of interest. So cloning site in the sense, here it will have restriction sites for the different enzymes. So in the tDNA region, so we need to have a cloning site, so flanked by the left border and the right border. So this picture shows the PBIN 19 vector. So that was the earlier made, one of the earliest made binary vector, that is a PBIN 19, that was made by Bhavan in the year 1984. After that, many modifications were made in those vectors to expand their utility as well as to improve their transformation efficiency. So like we do have the PZP 100 series of vector. So that is the smallest vector of size is about 6.636 kb size, and they are very stable in agrobacterium as well as easy to handle because of its smaller size. And binary vectors like the Picambia vector series as the widely used. They are constructed from the plasmid PZP with more choices of selection markers, namely with the NPT2 bar gene or HPT2 genes, as well as with the reporter genes like a GUS or green fluorescent protein. And these newer version of binary vectors, namely with the Picambia vectors, they have selection marker, that is a plant selection marker, very close to the left border. So that the last thing to be transferred into the plant cells, namely the plant selectable marker. So that the all plants selected for this particular selectable marker should have a complete tDNA region with our gene of interest. So there are super binary vectors also present, that is namely, for example, PHP 10525, so with the size of 552 kilobase pairs. So these super binary vectors contains the copies of VIR-B, VIR-G, VIR-C, and have made this agrobacterium mediated transformation possible with the monocots also, like rice, etc. So this binary bag, that is pi bag system, that can transfer up to 150 kb of DNA into the plant system. So here coming to the gene of interest, so that had come from any source, so that usually chimeric, that made from the parts of different genes. So this chimeric genes may be code for different characters or the different traits. It may code for insect resistance, disease resistance, or quality improvement. So coming to the promoter, so that is most important and that will drive the gene expression. So most commonly or widely used constitutive promoter is CAMB, cauliflower mosaic virus 35S promoter, and terminator. So that is also necessary in a gene construct. So that will terminator will terminate the gene transcription of that particular gene, namely NAS terminator, CAMB35S, and others. And we do have other elements that are present for the gene expression into the plants. So this is a electron micrograph of a cauliflower mosaic virus. So that has a circular double-stranded, that is a circular double-stranded genome virus. So coming to how this plant transmission could be done with using agrobacterium. So first we have to co-cultivate the engineered agrobacterium tumefaciens with gene of interest with an explant from which regenerated plants can be obtained. That means, for example, in case of rice, immature embryos or mature seed derived calli could be used as an explant to which we have to co-cultivate the agrobacterium tumefaciens containing our gene of interest. Then after co-cultivation, we have to culture the explant in a regeneration medium in the presence of a selective agent such as kanamycin if the tDNA contains NPT2 gene and an antibiotic that will kill the agrobacterium growth. So for to kill the agrobacterium growth, mostly the cefotaxime or carbonicillin or timitin will be used. So after the regeneration of the particular plant, so this plant is known as a transgenic plant, so that contains our gene of interest. So for that we have to test the transgenic plants for the presence as well as expression of the gene of interest or the introduced gene. And also we have to produce the progeny of the transgenic plant and we have to determine whether the introduced gene is heritable to future generations. And here the important point is, in case of agrobacterium mediated transmission, the process is somewhat inefficient. Only a small fraction of the target cells are transformed. And here the DNA must be integrated into a chromosome for stable transformation. And each transformed cell is unique in its character. So this slide depicts the agrobacterium mediated transformation of rice. So in that, as I told you earlier, the explant will be either mature seeds or the immature embryos. So from the mature seeds, we have to derive the calli, that is by inoculation in the medium containing M24D, that is mostly for the plant tissue culture. We will be using the Morozki and Skoog medium, or immature embryos, that is in the CC medium containing the 24D. So 2,4-D is an auxin that will induce the callus. And after callus induction, from either from mature seeds or from the immature embryos, this calli will be co-cultivated with the agrobacterium containing our gene of interest. That is, this is the strain name, namely with LBA 4404. So this co-cultivation will be done for 3 days in case of rice. So this period will vary depending upon the plant type. For example, rice it's 3 days, whereas for tobacco it is 2 days, etc. So these are all the chemicals added along during the co-cultivation process. So they will induce the virgins for the transfer of tDNA from the agrobacterium into the plant cells, namely 100 micromolar acetylsalicylate with 10 gram per liter of glucose at an acidic pH of 5.2 and with a temperature of 28 degrees centigrade. Then after 3 days of co-cultivation, we have to wash the calli with the sterile distilled water with 10 to 15 times to remove the agrobacterium. Then these calli will be kept in a medium containing a selectable marker, namely with 50 mg per liter of hygromycin as well as 500 mg per liter of cefotaxime. So why we are using cefotaxime means, so that will kill the agrobacterium. Then that will go to different selections, namely with up to third selections, 3 selections. So in all these selections, we will be selecting the calli transformed or the calli using the 50 mg per liter of hygromycin. So after selection, after third selection, then that will be transferred into a shoot-inducing medium. So this shoot-inducing medium will have the cytokinin, namely BAP 2.5 to 3 mg per liter. Then after shooting, then that will be transferred to a rooting media. So again, here also we are imposing a selection pressure using 30 mg per liter of hygromycin. So normally for rooting, we will be using a small quantity of auxins. Then after rooting, these regenerated, putative regenerated transgenic plants will be greenhouse conditions. And These plants will be tested for the presence or absence of transgenes by using polymerase chain reaction or by using ELISA. So till now we have seen indirect gene transfer method by using agrobacterium mediated transformation. Now we are going to see what are the different direct gene transfer methods available. So here we are having so many methods like micro injection, macro injection, PEG mediated transformation, electroporation, uh, liposome mediated transformation, microparticle bombardment, etc. So among this, the microparticle bombardment or ballistic transformation or gene gun is the widely used direct gene transfer method. In case of ballistic transformation or particle gun method, we are mixing the DNA that contains gene of interest with the microscopic particles of either tungsten or gold with the size of 1 to 10 micrometer, and the DNA will be precipitated onto the particle, and the DNA coated particles are placed on the end of a larger plastic bullet. The bullet will be loaded onto a gun barrel, and the target plant tissue that we want to transform is positioned at the end of the barrel, and the gun will be fired accelerating the bullet to the end of the barrel. So that's why the name is gene gun. And the plate with a small hole in the end stops the plastic bullet, and the small DNA coated particles maintain a momentum and pass through the hole and strike the target tissue. So this is the picture of the gene gun, that is the biorad that we are having in our CPMB in lab number 208. So in that, so we are keeping our target tissue in this chamber, and whereas our gene of interest will be placed here on a plastic thing. So here in between we will be having a barrier, that is a sieve, so that only the small particles, I mean DNA coated gold or tungsten particle will pass through this and will hit the target tissue. So in order to accelerate that, we are using the helium gas and by using a helium cylinder. So in this slide, it shows the inner structure of the gene gun. So this is the gas acceleration tube through which the helium gas will pass through, below which it is having a rupture disc, that is a plastic device that will rupture at a particular pressure. Namely, in case of rice, it need a pressure of about 1200 psi. So after reaching the 1200 psi, the rupture disc will rupture and release the helium gas at that particular pressure. So below which we are having a macro carrier. So this macro carrier is again a plastic device over which we have coated the micro carrier. So micro carrier is again, it is a gold coated DNA particle that is known as a microcarriers. Together that is known as a microcarrier launch assembly. So below the microcarrier we are having a stopping screen that is made up of stainless steel. So when the helium gas hit upon the rupture disc will rupture, and in turn with that high pressure helium gas will hit upon the macrocarrier carrying the microcarrier. So with that speed, that the helium gas will carry the DNA particles towards the target tissues. So here we have kept the target cells over the target shelf. So this DNA coated microcarriers will go and randomly integrate into the target cells or the target tissues. So again this target cells may be leaf disc or calli tissue or immature embryos. So after ballistic transformation, some of the particles will pass through the cell wall and enter into the individual cells. So some of the DNA will be released from the particle and end up in the nucleus and integrated in the chromosome, resulting in transformation. Till now, we don't know how it will integrate into the chromosome. So here in case of ballistic gene transfer, when compared to agrobacterium mediated gene transfer method, the multiple copies of transgene can lead to silencing. Here the chances of integrating multiple copies of our gene of interest may integrate into the genome. So here thereby it leads to gene silencing. Again, this consumables used for gene gun experiment are very expensive, and every time we have to import these consumables from that particular company. So coming to the important points in case of biologistic transformation, that is namely the direct gene transfer method, is it's having an unlimited host rate, because in agrobacterium in early days it was thought that it could infect only the dicots, whereas nowadays agrobacterium mediated transmission is carried out with monocots also. And here another important point is that the tissues or the target tissues or the target cell that doesn't have to be totipotent. And this is the picture showing the target cells, calli, and then regenerated calli tissue, that is with an example of rice, and then after that it will be transferred to rooting media, and it is in the jam battle for transfer into transgenic greenhouse conditions. Coming to the next direct gene transfer method, that is a PEG mediated transformation method. So here we are digesting the cells with cellulase to get the protoplast, and the PEG polyethylene glycol induces a reversible permeabilization of the plasma membrane. And here the important thing is it's the viability of the protoplasts are very low, as well as the transformation efficiency is very low, and it is very difficult to regenerate the plants from the protoplast. So these are all the isolated protoplast done with PEG mediated transformation. So in case of protoplast, we need to have an in vitro regeneration protocol, as well as in case of gene gun transformation, it may lead to somochronal changes. Again, that is a very much problem for the transgenic plants. Another important direct gene transfer method is liposome mediated transformation. So here the targeted DNA is encapsulated in a spherical lipid bilayer that is termed as a liposome. So in the presence of polyethylene glycol, endocytosis occurs. So after endocytosis, the DNA is free to recombine and integrate with the host genome. Another method is electroporation. So here we are giving intensive electrical field that will lead to pores on the plasma membrane, allowing the DNA to enter. Again here also through electroporation also protoplast regeneration is a problem. And we can also directly transfer DNA using silicon carbide fibers. So these silicon carbide fibers are used to punch holes through the cultured plant cells. The cultured plant cells are added to a tube and vortexed vigorously. The mechanical force generated by the vortex drives the fibers into the cell. Another one is micro injection. We are using fine glass needles to inject the foreign DNA directly into the host cell, and it is developed to inject DNA into protoplast as well as the cultured embryonic cell suspension as well as in the multicellular structures. But this micro injection is a very time consuming procedure. So with that, we are end up with the gene transfer methods in plants. So to sum up, we are having two gene transfer methods or a genetic transformation from plants, that is by direct gene transfer, indirect gene transfer. In the indirect gene transfer method, we are having only one method, namely with agrobacterium mediated transformation. Whereas direct gene transfer method, we are having so many methods like micro injection, macro injection, liposome mediated transformation, PEG mediated transformation, silicon carbide fiber, or gene gun or ballistic transformation. So among all these direct gene transfer method, ballistic transformation method is the widely used method. Thank you all.